You're happy it's over. Um, there will be, like I said last time, right? Next week, Wednesday, there'll be a Q&A lecture, so you can come and we'll ask or we'll answer, not ask any and all questions that you ask. And so we've sort of run through all of the materials that we wanted to go through. I think you have a nice overview of what fMRI does, its pitfalls, but also its potential. And since I'm the researcher of the two that does most in terms of fMRI, I thought it'd be nice to sort of close off with the sort of just the scientific talk that I give when I give an overview of what my lab is doing. Because now I hope that you're all at the level where you can sort of follow anything that we do. And I hope it also gives you an idea of, what is on the forefront. Not that I necessarily, that my research is at the forefront, but it's the sort of things that we're discovering that I'm very enthusiastic about that I would love to also show you guys. A few other household things. I've always said that I wasn't going to put up any recordings, but I did secretly record all of the lectures. I haven't checked whether they're all successful, but I will be converting them after the weekend, and I will put them on YouTube so you can go through all of the lectures for studying if you want. So I haven't checked whether everything is recorded well, but I did secretly record them, so I'll put as many as possible online and link to it in an announcement on Canvas. Yeah. Sorry for lying to you. Yeah. Yeah, what else? Oh yeah. It is part of the exam materials in that it's in where it sort of draws links between two things that you already know. But most of the things that I will ask during the exam that have anything to do with this lecture will be more part of the bonus materials, so bonus questions. So this is also not quizzed for this reason. It sort of stands outside the core course material. Any other questions?
Is the bonus material on the exam, is that purely bonus on top of like standard?
Yeah, so you can get a grade 10 if you don't make any mistakes during the course. And you can up that to, I believe, at 11.5 maximally if you answer the bonus questions on the exam perfectly also. So I have seen people score like a 10.3, and I'm very sorry to say that I then have to round it down to a 10 for those of you that are very ambitious. Yeah, so that's the status of the bonus materials. Yeah. Are you all reading the last paper on Purusol? Great. You'll see that the approach that they are highlighting there lines up a bit with what I'll be talking about today. So there'll be some back and forth perhaps between those. Cool. This is a lot of material. And so I'll have perhaps a tendency to go through it pretty quickly. I'm trying to show restraint in that sense. If you have any questions, just interrupt me, and I might skip certain things that are then also definitely not part of the quiz material. There's already a question in the chat. Yes, I'm working on this document of a list of caveats. I'm at number 8 now. I'm anticipating I'll go up to something like 25, but I have started writing that. I hope to also finish that around the time of the Q&A next week. Clear? I might even publish it, so thank you all. Great, so let's go. one of the core themes that we are sort of finding in the lab has to do with these sensory maps that I've already shown you. So this is the lab that I run is both at the VU and at the Spinoza Center, and then it's also a bit at the NIN. So the NIN and the Spinoza Center are at the AIC, right, at the hospital campus. And both of these are then part of the Royal Academy of Sciences, but that's just I also give this talk at other institutes, and they always want to know this sort of thing. I don't really know why, but yeah. And so the core concept that you'll be seeing recur over and over is this idea that if you look at the cerebral cortex and its sort of major mode of organization, it's that you have these maps on the surface of the neocortex. And we know from like ages ago that this is how sensory motor cortex is organized, right? You've seen this in the neuroscience lecture. We also have these visual maps in the back of the brain. We have phonotopic maps. You've seen examples of those too for audition. And you've also already seen that these maps actually exist for not just sensory qualities, but also for more abstract, more high-level cognitive types of categories, right? So these are the numerosity maps that colleagues at the Spinoza Center have discovered. So just to highlight this thing that I really like about this research that my colleagues did is that if you zoom into this parietal lobe region that has this map, you see that it has this numerosity map that's organized a bit like this, a bit rotated relative to this, but this is what that map looks like if you zoom in. So that's nice. It's a numerosity map in parietal lobe. But it turns out that if you run the same subject, not just in a numerosity experiment, but you also do what we call a retinotopic mapping experiment, where you map their visual field maps, you actually find structured activations in the same region. And so what I can do is go from this numerosity map in this patch of cortex and flip to its visual map. So what this means is that we can find all these sorts of maps in the brain, and these maps, they work together. They sometimes overlap in weird ways. And this to me is a sort of very nice sort of clue as to not just how the brain is organized fundamentally, but also to how the brain might sort of fuse different types of information processing together by overlapping these maps. And finding overlapping maps, though, then means there is some sort of possible interaction between two modes of processing that we then have a reference frame to try to start to understand. Is that clear? Right, so in this case, it might have something to do with how visual processing interacts with numerosity processing, right? And this interaction could occur locally in these regions of the brain. And that's an interesting, compelling sort of thing. Now, if we start looking at the interaction between visual and numerosity-based processing in that region, we can start to get at the mechanisms. Yeah, and so we're part of the NIN and the Spinoza Center. And this is also something that you've already seen, right? So this Hubel and Weasel movie. Way in which the brain is organized helps it compute. Now, our hope is that if we use the scanner well, we can start to really move towards the level of understanding the living human brain by using scanners to look at local neural populations, like what are local neural populations, like maybe a couple of thousands of neurons doing together. And that's what we're hoping we are measuring using fMRI. If we use the 7 Tesla scanner, for example, because of its higher SNR, we can have better SNR and smaller voxels, be more specific, be more sensitive. So this also allows us to look at single trial responses. And So you have this sort of, I have this sort of feeling that as fMRI as a field is moving towards using scanners like this, it's really starting to break like a glass ceiling where we're actually starting to be able to use it as a like a hardcore scientific measurement thing. And we're trying to move away from like blobology by being able to look at the whole brain at once and still be able to find certain computational principles, right? And these two things that I've already highlighted, this idea of the receptive field and this idea of the maps in the brain are these types of things, computational motifs that we're interested in how they underpin cognition. So I once gave this talk in Marseille. This is the old port of Marseille. The idea is that we'll take that concept of the neuron, as I explained in the encoding models lecture, right? And then say we're going to pretend, or at least like assume, like we always make these assumptions when we do fMRI. Our assumption now is that when we take a voxel, it actually has a neural population inside, and we can sort of take a measurement of that and then pretend that that's the receptive field of a population of neurons. That's population receptive field model. You know this model, of course, because we talked about it. So different voxels have a different viewpoint on the outside world, where these dots are neurons, and these are these are neurons receptive fields, and we sort of sum them together inside a voxel, which gives us the population receptive field. So that's the receptive field of the population of the neurons. We sort of see this as every voxel having its own little sort of camera on the outside world. Yeah, it's localized periscope sort of thing. So one other thing that we do in the lab that I think is important to highlight is this idea that we have these models that try to explain what's going on in this single voxel. So we don't just pretend that we're looking at a population of neurons that we can characterize, but we actually use specific computational models to characterize what these populations of neurons are doing. So what we generally do is we have a computational model that we put in a stimulus and a set of parameters. And the parameters then serve to tune what the model does with its processing of a stimulus. We make a prediction of what the time course that we find in a given voxel should look like, given some stimulus and given some parameters, and then we fit that to brain data, right? So we fit this computational model to voxels responses throughout the brain. Yeah. So this is somewhat different from the standard GLM approach, where you have sort of a fixed model, right? And you just get beta waves throughout the brain. Here we're actually optimizing parameters. This gives us two specific outputs. We were very interested in those parameters because they, for example, relate to the position in the visual field, or the frequency of sound, or the number of items on the screen, or in the case of these semantic maps, right? A certain set of words that relate to one another. Right, so that's what's given here. And we can also see how well we explain our data by looking at prediction performance. That is, if we fit the model on one data set and then try to predict in another data set what the time course should be like if our model has fit the data. Okay? So this is very standard in machine learning. Like, it's very machine learning type approach. And that's what we're trying to do here. So the idea is that by fitting these parameters, we can start to inspect what they mean in computational terms. And that allows us to really probe how the brain is representing information. It's these, for example, these word clouds, when you click a voxel in the semantic processing, right? You get this word cloud. That's what that voxel represents. On the other hand, if you have multiple different computational models, you can start comparing cross-validated prediction performance. And that's a very important way of sort of validating what type of computational principle is the brain implementing. Yeah, is it this one or that one? Well, this measure allows you to say which one is the best of the two options. Yeah, so it's kind of different from hypothesis testing, but you're letting different models compete to explain the data. It's a bit like a ImageNet sort of thing or something. Now I'm going to quickly go through this. You've seen this before, right? So we have this model. We can tune the parameters to best explain a voxel time course. So in this experiment, we have 8 of these bar passes, and we can fit for each voxel what part of the screen it likes best, right? Where it activates when a visual stimulus is there. So we move these things around the location and the size, different parameters to best explain a voxel's time course. And if we do that per voxel, then we can project everything back into the brain, right? And thereby inspect, okay, what is the information processing like throughout the brain? And by having this information from each voxel, so we can say, this voxel likes something that's close to the upper visual meridian. So the vertical meridian seems to happen at the top of the screen. It relates to the screen. So this is the way that we find these maps, by trying to explain single voxel responses. Yeah, this is all recap, of course, from what we've talked about primarily in the encoding model lecture. So any question about this? Because this is none of my work, mostly. Very clear? I was really hoping I would have like 10 seconds to have a sip of coffee. No questions? Good. Then I'll just assume that this is all clear. So then, the spiel of what we've been doing in the last 5 years or so has been to really try to see whether we can take these concepts further. And one of the ways in which we've tried to do this was to see whether, so this is a pretty conservative field, this field that does this sort of mapping. And most of these people are geeky AF, and they just care about these slight differences between, like, LO1 and LO2 might be different, right? And that's interesting, but it is a very sort of narrow scope. And what we've tried to do is broaden our scope a bit and see, can we find this type of visual response elsewhere, right? Because people have been partitioning the brain into like a purely visual part, and a purely auditory part, and a purely, I don't know, reinforcement learning part, right? And our idea is really that if these maps that we find, if they multiplex, and that gives our brains the ability to fuse different types of information processing, then maybe there's visual maps in more locations, right? If vision needs to interact with reinforcement learning, maybe there would be a map somewhere else, right? Instead of just these core visual regions, we're going to try to find this type of organization throughout the brain. And so, yeah, that's what we did. So we tried to take this huge data set. It's from this Human Connectome project that I've talked about before. It has this specific retinotopy experiment, very similar to the bar thing that I showed you just before. And it's an insane data set because it's the biggest in the world. It's an American data set, so you get that, right? It was acquired in Minnesota, not Texas, but still. And what you get is you can average 180 subjects together if you want insane signal to noise. And I've taught you that you shouldn't average subjects together. Now, there's two reasons why we tried it here. Anyway, one is this is that HCP data set that is overlaid best, so we just thought we would try. And the other is that for the subcortex, it works better than for the cerebral cortex, like we went into in the comparisons lecture. So our hope then is that we get something back from averaging so many subjects, because an order of magnitude more than you can get for a standard endpoint. And then look at whether voxels show specific responses. And it turns out that yes, when you do this, you can start to find these visual maps that we know from the visual system, like very closely related to the retina. We actually find them in cerebellum. And this is this region that we thought that traditionally is always seen as a set of brain regions that does motor control. And so we didn't just find like a single voxel that did something, but there's actually a lot of structure there. And this is also why I really like this idea of these maps. If you find a blob, well, that's great, right? But if you find a map, that means that there is like an inherent sort of structure in that map, right? It reflects the organization of the sensory organ. Right? And the fact that that is retained, that structure, is very informative, right? It also guards against things like null findings. It's not just a blob. It actually has an organization that you can then look into. which what we did. It turns then out that you get like not just one or two regions, but you get three of these clusters, and there's actually 5 different visual field maps inside cerebellum. And each of these maps turned out also to have a slightly different organization that would fit better with specific function. I won't go into that in detail now, but we went into that in the paper. So if you're interested, you can look it up. One of the things that we think happens here is, hey, this is this fusion between vision on the one hand, right? And we didn't know this before, but now we see these visual responses there, and they're very structured. And it has to have something to do with, people didn't think that this was a motor region for nothing, right? This is a very motor related region. So what we think happens here is that, well, we know this from the literature, this new retinotopy, it actually overlaps with body map representations that we know exist in the cerebellum tradition. So if you look at the flat map of the cerebellum, so you can actually fold it out exactly like you would the cerebral cortex, and it looks like this alien is staring you in the face. And it turns out that you have these body maps here and here in the cerebellum and over there. And these white lines, they circumscribe these new regions that we found. So it's actually like 20% of the cerebellum actually is visually organized, and they're outlined in these white things. And you see that the bottom ones actually overlap with these body maps. And so that seems as if we have this multiplexing thing going on there, where the visual map overlaps with the lower body, it seems, mostly. So that's kind of funky. And of course, we can't have people walk around in the scanner because, yeah, it's pretty shitty. But this is what we get by overlapping this with the literature, this finding. Yeah.
What would they think functions this? That is maybe hand-eye coordination.
Exactly. So we think that, yeah, this is one of the two things that we think happens here, right? So visually guided behavior, right? When you pick up something, you need to fine tune how you're moving your fingers when you're juggling. Yeah. But like, this is a very beautiful finding from a paper that came out like last year. This is the first paper where what they did is they had people walk around with a portable eye tracker in real life, right? So this is, this is the sort of environment that we evolved to navigate, right? And what you do all the time is you move your head around, you move your eyes around, and you plan where you're going to place your foot, for example, right? But then you need to remember where that rock that you're going to place your foot on, where it was, right? And you have to implement some sort of fusion between the visual information processing that you just went through when you looked at the rock and what your foot is going to do, for example, right? So this is one of those ideas where if we hope that if we look in detail at these interactions between the vision and the movement parts or activations in the cerebellum, we would find interactions that then have a relation to how this type of behavior and cognition works. Yeah. Yeah. So you can't have people, let's say you have people walk around in the scanner, they'd be on their back and they'd be like all moving around, right? So that's horrible. You can't do that. But what we can do is do like very controlled movements. So we've actually done an experiment where we have people like clench their toes, for example, and you can remain pretty still while you're doing that. And then you can have people move just the thumb and then move just the fingers, like index fingers. So you can go through the entire body. So you can also have people like raise their eyebrows in the scanner or pouch their lips or whatever. And so you can go through a whole set of sequences, sequence of movements, and then do like a GLM analysis and find these body maps. Actually, this is project 2 in the extra notebook. So if you're interested in this sort of thing, you can actually, the end result of that project is actually this type of map. You can actually find the finger map in motor cortex in that project. Yeah, that's good. That's a good question. So it does turn out that your SNR, your signal quality in the cerebellum is generally very low, but you can image it. And so in this project, they did image it. It's pretty state of the art and all of that. So normally what people do is they say, yeah, well, I don't have a state-of-the-art scanner and I do want to push everything. So what I'm just going to do is zoom into the cerebral cortex, and the cerebellum is right on the bottom there. And so they sort of cut it off. That happens a lot. So we are blind to the cerebellum because of that. Yeah, so my colleague Wietske, she's running the MR physics lab in the Spinoza Center. And her whole game is to optimize scanning for the cerebellum. And the idea then is that if we can get better at scanning the cerebellum, that also helps scanning the rest of the brain. Just trying to push the envelope a bit. Yeah. Awesome. Yeah. So this is just an example of how we think we can start to find this sort of more multiplexed cognition type things. It turned out that when you go into this even more deeply, that we do the same analysis and we find retinotopic maps even in the hippocampus. So I told you about the hierarchy of visual processing, right? It starts with the retina, it ends up with the hippocampus. Well, people always think that in the hippocampus, we have these cells, they report where an agent or an animal is in the environment. So that's completely abstracted from sensory coordinates. And what we're finding here is that there are actually sensory representations also in hippocampus, which I think is pretty compelling, right? Similar to the cerebellum finding, whenever we use memory to function in the world, that means that the memory has to interact with the information that's coming in, right? So it makes sense that these regions that do the most complex and abstract information processing, that at some point have to grasp back to the sensory coordinates, right? In order to, for example, form a prediction of what's going to happen, right? If I'm stepping into a room and I know which room it is, I also know it's likely that certain people will be in there. So when I look at someone's face, I can choose from a set of people that are most likely to be that person that I'm looking at that moment. That means that that memory has to interact with very specific sensory information processing. Yeah, so we published this in a couple of papers now, and what you see here really is also that, like, for example, on the left here, especially here, you see that it's more blue, right? The contralateral field of view, you see that it's more red and orange there. Yeah, so it really means that the left side of the screen is represented by the right hemisphere, as it was in V1. It's also in hippocampus. And what I found really cool is that usually you trace, like as a fMRI researcher, you go to the animal literature, right? And you see, they found this in an animal, like for example, for the numerosity maps, people have found in monkeys numerosity neurons. And then my colleagues thought, okay, let's try to find this in fMRI. But here it worked the other way around. So all of the animal studies were still doing this environmentally oriented receptive field mapping in the hippocampus. And only after our work came out, I'm just sort of blowing my own horn here, sorry. People found that the same type of thing, they sort of replicated this finding in human fMRI, replicated it in the rat afterwards. So it's a really cool sort of interaction here, right? So you see that you're actually doing research that maps onto animal research. You're really sort of mutually reinforcing. Yeah. So why, right? I want to sort of loop back onto why this would be the case. Like I introduced it a bit, like saying like these multiplexing of maps and that sort of thing. That's all good. But we can sort of develop a bit more structured thought about why these maps would be important to look at. And so one of the reasons there is that if you have multiple regions in the brain that all are organized according to the example here, vision, right? It means that you can set up information processing that follows this structure, right? And that would be very, very efficient because whenever an object lands on a specific portion of the retina, all of these regions are working together to analyze that specific information on that location of the retina. And so what this means is that you can have a lot of back and forth between regions that are organized in the same way, and it can start to function as a sort of common currency for different regions in the brain to communicate. Or you could call it a common language that they all sort of speak. The other idea that I think is nice to point at here is that this thing that I also highlighted with the hippocampus example, right? We always interact with the world, right? We are stuck in this, our brains are in a vat, right? And the only interface we have with the world is through the sensory apparatuses that we have. It's the surface of our skins, the posture of our muscles, the visual information coming in, all of those is what we might call the sensorium. It just means that this is our window onto the world. All of these things together are how we interact with the world. And all of our behaviors, even if they're very abstract, they have to run through these sensory impressions. They're always in touch with these sensory impressions. So it also is a way in which the brain can interact with the outside world. So it's not just the inside of the brain where this sort of principle would work. Also, its interactions with the outside of the world. Well, the outside of the world, the outside world. Yeah, I'm just going to close off this part. Any questions? No? Cool. So this is all nice and good, of course, but it sort of goes against this idea that, for example, hippocampus is very abstract in its information processing. Yeah? Looks like I'm saying it's just low-level stuff, right? Circles all the way up. But that's not really the case. We know that information processing is becoming more and more abstract as we, for example, go from V1 to the hippocampus. So we do know that there's abstraction going on when you go from the retina to more high-level regions in the brain. So this is a diagram of that same visual hierarchy that we sort of recreate in convolutional neural networks for vision, for example. Here you see that structure. Well, these are very analogous, and we know that information processing, as we move up, becomes more and more complex and more nonlinear and more position invariant, for example, more abstract. And so one thing that we did is we went to these computational models to see whether we can model this process of increased nonlinearity of responses and position invariance. So we try to focus on these very basic computational principles. And this is actually work done mostly by Marco, who's a PhD student. He's now running the psilocybin experiment. And this is all done in Serge's lab, right? So Serge and I collaborate a lot. He's the director of the Spinoza Center. And so the idea here is that we can create a population receptive field model. So that's one of those models where you just look at the voxel responses and you try to explain them using these computational models. Right? We're going to take that simple model, and we just have a blob in visual space that we think the voxel responds to. And we're going to try to make it a bit more complex, see whether we can model things like this position invariance and like the nonlinearities that we know happen as you go up the hierarchy of processing. We want to try to understand that evolution of increased abstraction in the brain as a function of these basic computational principles. So the question really is, how does the brain create this invariance from these sensory coordinates, for example? And what we have now, generally, is the simplest PRF implementation is what happens is we just have this model, right? So this is that position and size of the model, and we just multiply it by a stimulus, and that creates our response. Very simple. But there's something wrong with this in that it's just a multiplication. So it will go on to infinity. For example, it will not be able to capture responses that slightly compress, right, that don't go to infinity. You can imagine that if you show a stronger and stronger stimulus, at some point you would expect a bold response of 300% signal change, and that just doesn't happen, right? The brain would explode, and it doesn't because it has this sort of tuning where it goes down, it compresses things. And the idea here is that we can make that happen by this process called divisive normalization or divisive normalization. And this is a process that's been theorized for a long time. Again, I won't go into this in the exam, right? I'm just showing you how we're doing this sort of thing in the lab. But the idea is that you have an input that leads to a response like it would here, but it's not the only thing that's being done here. What's also happening is you have other inputs like neighboring neurons. And what the neighboring neurons are doing is they're saying, I'm also active, right? And so you're my neighbor, you shouldn't be so active now, right? Because I'm also active. And so what the specific implementation is that other inputs that are similar, they are actually summed together and they're used to divide the response here. You can imagine that if things are, so this is a beautiful the computational principle, which we go into in the neural models course in depth. So I won't go into it too much now. But what we've been able to do is say, okay, I'm going to make a PRF implementation that does exactly this. It's very simple. We have an activation thing. That's just a thing that you just saw. We also have a normalization pool that's just the same PRF, but much bigger. These are the neighboring neurons, right? They are right next to what's happening here, but they're like a pool of neurons that surround it. Okay? And we can do the same thing. We can just sum all of those up here and then say, I'm going to divide by the thing that's in the bottom of the fraction here. Okay? So that's the main, very simple computation that we're performing here. And the nice thing is that this leads to very interpretable parameters. So this B is how strong the response would be if there were no stimulus on the screen. This D is the sort of baseline activation of that normalization pool. Whenever there's no stimulus, you do have B and you do have, you have B and you have B. So these parameters we think we can interpret in biological terms. And it turns also out that they both have specific signatures that they give, right? So if you, for example, if you go and you increase B to the value of 100 from 0, so what you're doing is you're changing that value here, very simple thing that you're doing in that model. And what happens is it starts to gain surround suppression, which we know is very important in, we've seen this in neurons, and we also see it in fMRI responses. And conversely, that D parameter here at the bottom, what it does is if it is smaller, it will do this response compression. It will compress the response to a certain maximal value. So disallowing it to keep going and rising to infinity. And so you see how we have a very simple model. In principle, you don't have to necessarily understand all the ins and outs here, but we have this very simple model. It's just the same model replicated, and there's a few parameters that really produce very distinct response signatures that we can then start to look at. Well, it turns out that when you do this cross-validated model comparison, yeah, go ahead.
Model, I was wondering if this model similar with how we develop resistance to everything.
Resistance.
Similar,
What do you mean by resistance? You mean like how your brain or your body adapts to things that have happened in the past? Mm-hmm. Yeah, it's a good question. So it turns out that we think, like people other than us have thought that this divisive normalization is a general principle, and that what we're using here is a spatial incarnation of that idea, right? So of a surround, literally a spatial surround. But we think that many sort of types of processing that evolve over time, they actually also have a surround, but there it's a wider range of timescales, right? It's a slower developing response. And so you can also use what has happened in recent history to divide what's happening now, right? It's the same thing. And so this principle, just the general principle, also explains things like adaptation, right? So the beep that you hear after an explosion, like the sort of thing, all that sort of stuff is also explained very parsimoniously by this general computational principle.
But it also then explains something like the resistance to certain drugs.
Mm-hmm. Yeah, in the same way, right? So if you, so if you have been exposed to something in the past, you're gonna need more, right, to get the same effect right now, because what you're inputting now is being divided by what you had before. So that, because that is now something instead of nothing, you need more of the drug to get the same effect. It's also the reason why people overdose after rehab, right? Their recent history, they haven't had any heroin, right? And they just take the same amount that they're used to, like from months back before rehab. And then it has much more effect than it would have had they been still addicted, something like that. You get the point, right? Cool. So it turns out that when you do this like cross-validated model comparison, this model is better than all of the competing models, where certain models can only do the suppression thing on the flanks, right? And others can do only this compression thing, right? So not rising to the peak. And this happens like all throughout the visual system. And to quickly cap this, you can then take all of these specific visual regions as regions of interest, right? Just select everything that's going on like there. And then you can see that this B goes down from V1 to higher visual regions, right? So this is a signature then of increased abstraction, right? And similarly, something happens with this D, it also goes down. Also, this evidence of this increased abstraction, increased invariance that happens when you go from low levels of processing to higher levels of processing, yeah? And so what this does is it allows us to reuse that that retinotopy, right? That spatial coordinates of the sensory inputs, and try to understand this process of abstraction as it happens, going from low levels to high levels. So Marco is now doing a psilocybin study. He's also looked at PET receptor density measurements. They all correspond beautifully to these parameters, right? So we're really trying to start to build the case that these parameters are actually biologically relevant. We have a very, very specific prediction also as to what should be happening in this model with which parameters when people are on psilocybin. So that's what we're working on right now. For those of you that are going to be a subject, now you know what you're supposed to be doing, what your brain's supposed to be doing. No, that's, you don't. Oh yeah, so there's a question here whether the idea is that visual maps are this common frame of reference, or whether there are other types of maps that could also be a common frame of reference. And I think it's a general principle. And at the end of the lecture, I'll show you why I think that. Okay, let's have a break. I don't know, till 55 on that clock. That's always goes wrong, I know, but you know, I just keep saying it. Yeah. See you guys back in 8 minutes or so. Mhm. Oh yeah, so I took it out. So the surround is in V1, it's like 8 times the size of the center, right? So the standard size of the center is like 1 degree or something in V1. So that means that in the surround is like 8 degrees. Yeah, it's large, right? So it's like even a voxel that has a center here will actually have a normalization field here. It will extend all the way there. Yeah, so it's a parameter in the model, right? So it's just optimized in the fitting procedure. Yeah. But of course, you know, if they're very large, then you only have a stimulus this big. And so it becomes a bit uncertain what the exact value are, because you can't really hit it with a stimulus that well. So we're working on a bit of that, but yeah. So this ratio, what I said, 8, right, is the ratio in V1, and it goes down to the level of 5, 4 if you're in like more high level visual regions. So this surround actually becomes more specific relative to the center as you go up the hierarchy, which we think is also. Yeah. Yeah, so we think that we're onto something here, like as a general principle. We have some ideas on how this goes even further. But it's very hard to make sense of. Yeah, I'm still breaking my brain about. Ah, yeah. Mhm. Mhm. Yeah, nice idea. Yeah. Yeah, I don't know. So yeah, this would be necessary for the brain to overlay vision with sounds, for example, right? Because sounds are fixed to the head, and vision, we move our eyes, right? So you need to, like, at some point create, well, not necessarily create, but find a way, the brain needs to find a way to overlay vision, right? And it needs to use eye position for that. That's true. Yeah. Mhm. Yeah, so I don't know of any literature that has explicitly tried to map out sort of craniotopic space with sounds. But there is a paper, Nature Neuroscience 2007 or something, where in Clay Curtis's lab, do you know Clayton Curtis from the working memory stuff? He actually has a paper where they show that there are responses in frontal eye field for stimuli that are behind the head, so auditory stimuli that are behind the head. And so they say, like, that you actually have like a more world-like map there. I don't know if that's, yeah, well, that's a nice finding, but there is not much in terms of like specifically mapping out the whole peripersonal space with audition. Yeah, that would be really cool. Yeah. Mhm.
Because it is not what level it's actually not.
Oh, yeah. So we do have a, I can show you here. We have a paper that we're going to submit in what is it? Like, hopefully soon.
Is it the one with Ines? No,
Yeah,
it's not.
Ines is on there also.
Oh,
No,
okay.
but Ines is a co-author, but this paper is not. And so what we've done here is we've tried to dissociate the visual space from the screen space. And so what we've done is we've done like PRF mapping at the center is in this circle, right, of the screen. So the
Yeah.
black is the entire screen. And then we've moved gaze, right? So we do the entire experiment again on the left side of the screen or on the right side of the screen.
Okay.
You see that there's zero overlap between what's being hit by the stimulus on the screen here and what's being hit by the stimulus on the screen here. And so the question is, like, if the receptive fields that we find are all screen coordinates,
Yeah.
then we should find completely different responses here, right? Whereas if they're fixed to the retina, we should find the same throughout. So we have a very strict hypothesis, and it turns out that yeah, it's all, you have a retinotopic prediction and a spatiotopic prediction, and it deteriorates very rapidly when you have a spatiotopic prediction, so meaning it's retinotopic. And this is something, and we also have an attentional condition, it's not very important. And then we can do the same in a bunch of analyses. We keep showing the result is always that we have retinotopic prediction that things are not overlapping spatiotopic that they should be. You see a wide difference there. And so throughout the visual hierarchy, we keep seeing, this is very slow. So we can also decode like stimulus position from the brain activation pattern. And then you also see that it's all retinotopic. So spatiotopic would be here, it will be all the way on the left, and you see that everything is on the right.
That's interesting.
Yeah.
So because I guess spatiotopic in your experiment is also body topic.
Yeah.
Way, because you're
Yeah,
still. Right?
your head's fixed here.
So it's really moving with your eyes.
Yeah, it's really fixed here right
Okay,
now,
not
in our
body.
case. Yeah.
Okay.
Yeah,
Interesting.
so that, but this is a very contentious point. So there's a lot of discussion in the literature about this. And so, yeah, we put a lot of work into this. I don't know if people will buy this. We still have to. Yeah.
Good luck.
Yeah, thanks. We'll need it.
Thanks.
Yeah.
Je kunt buiten stukken aan
So, there's more, of course. What I want to loop back to is, this idea of topographies, right? We've just been looking at how we explain what's happening in a single voxel. And we talked about maps. But now I want to use the maps to try to understand functional connectivity, right? So we talked about this in the previous lecture. Functional connectivity is trying to understand what a given voxel is doing as a function of what's happening in the rest of the brain. Yeah. And so can we use this perspective on maps? So kind of give maps their rightful place or something? Can we use that perspective of maps to understand functional connectivity? And so I've already talked to you about this. I don't know if you realize, but what this is a very, very strong hypothesis. This is that first region, right? That's common language between regions in the brain. If they can all speak vision, right, they can talk to one another. And what this means is that if they're all talking to one another in this language of vision, this gives us a very strong prediction about how their connectivity, in terms of their co-fluctuations, how that should be organized. People have been thinking that this is the case for a long time, but they haven't been so focused on these sensory topographies, and that's why we thought this would be a nice thing to try. Right? So if you show a single visual image, this gives you all these retinotopic reflections in different visual regions, right? And we've just seen that these different visual regions, there are actually many more of them than we thought before. This actually makes it interesting to look at how this retinotopic connectivity is structured. Maybe we can start to understand what the brain is doing. And so our approach, because we are one-trick ponies, is that we're going to use this idea of the population receptive field idea, right? That model, trying to explain what happens in a given voxel as a function of what happens on the screen. What we're going to do now is use exactly the same modeling approach, but now replace the screen with another brain region. And so what this means is that so we just replaced receptive with connective, right? And now what we do is we have a model that models brain responses somewhere else in the brain as a function of some pattern on the surface of, in this case, V1, right? So that's the first visual region. So to be clear, we talked about seed-based connectivity 2 days ago, right? That is when you select a given voxel in the brain and you calculate what other voxels throughout the brain, how they correlate with that given voxel. Now, this is very, very similar. It's just that now we're not using a single voxel as a seed, right? We are using a specific spatial pattern, again, this Gaussian shape on the surface, as our seed. So it's very simple. And this gives us a computational model that we can use to understand functional connectivity. And the nice thing is we are sort of using the structure that we know is in V1, namely that it has this retinotopic map. This gives us a computational model that has two important parameters. One is, what is the best fitting peak of that location or of that patch, right? So we can move it all the way there, all the way there, all the way there. We can move it around. That's what our fitting procedure does. We can also make it larger or smaller, right? If the functional connectivity of some voxel somewhere in the brain is very specifically connected to some patch in V1, then the size of this model is very small. It's a very, very specific patch on the surface of V1. And so we can do exactly the same thing that we've done over and over. We're actually able to fit our receptive field model type thing to brain responses. And this was the trick that I've always been doing, that I've already been doing, that I thought was working. So now we're just going to keep doing that. It's just that now we can do it inside the brain. And this has several very important benefits that I'll get into in a bit. So one of the things that this allows us to do is that if we find this sort of what we might call topographic connectivity, right? So that's different from just functional connectivity. It means that the connectivity is shaped by the topography of the source region, right? In this case, V1. And we can try to see if we can use this connected field modeling to retrieve these known retinotopic maps. Because what this method allows us to do is really like project V1's retinotopic map into the rest of the brain. What I mean by that is, so this is V1, right? It's between this red line and that cyan line. If a certain voxel in the brain, somewhere in frontal cortex, likes this yellow, right, likes this patch in V1, that's when it's preferentially connected to, then we can just give it the yellow color, right? And if the voxel next to that voxel in frontal cortex likes this patch in V1, we can give it the green color, right? And so this means that in frontal cortex, we can just create these beautiful colored maps also, just by relating responses throughout the brain to the map of V1. This is why I say we can project the retinotopic map of V1 into the rest of the brain, finding these topographic maps. And the beauty of this is that we can also start to then explore this type of visual organization during natural vision. And what I mean by that is, I don't know if you know, but that receptive field retinotopic mapping experiment with the bar, boring, right? You had to like look at the center of a screen for an hour. That's totally unnatural, right? We don't do that. We look around. Moreover, this bar stimulus that we're using, it makes no sense, right? That's not like the real world. We are just looking at things like natural things, right? Things are objects, things are scenes, things are, I don't know what, right? Things are things, not just a bar, right? Bar filled with checkerboards. Like, that's not, that's not an accurate reflection of the world at all. But you want to look at the brain while it's doing something that it was meant to do, right? And these movies are actually very, very similar to, you know, what our brains are meant to do. It's active, you're interacting with the material in the movie, it means something. All of that is visual-based processing that the standard retinotopic mapping experiment is completely disregarding. Yeah? You're only looking at a very thin slice of cognitive, interesting vision when you're doing the retinotopic mapping experiment. Whereas a movie, you're actually engaged in the movie in so many different ways. That's really what the brain, what vision is for the brain. And so what's cool is, because this methodology is fixed to V1 instead of the screen, it doesn't matter that people are moving their eyes, because it's not V1 that's moving. You can imagine that if someone in the retinotopic mapping experiment moves their eyes, you're completely gone, right? If someone moves their eye, it shifts everything on the retina, and your complete model is off. You have to throw away the entire run when people do this. Okay? Here, it doesn't matter anymore. It's just part of it. You always have this connection to V1, because that's not changing. The nice thing is also that you can do it during resting state. Right? You can just look at whether, when you're just thinking of stuff, whether your brain activation is also organized according to this organization. And what's nice is this data set that we already knew how to use. I'm just a scavenger, right? This was during Corona times also. We couldn't do any experiments. And it just turned out that in this Human Connectome project, no one knew of this data set other than the retinotopic mapping experiment. It turned out that all of these 180 subjects had not just done half an hour of retinotopic mapping experiment, they had actually also done an hour of resting state scanning and an hour of movie watching in the same subject. So we could compare the retinotopic mapping experiment with the movie watching experiment with the resting state, and then see whether at the subject level this would align, give us the same maps, and so forth and so forth. And no one knew about this movie watching data set, for example. The people that did the project, they just forgot to publish a paper about the movie data set. And so for a while, we were more or less the only people in the world that knew how to find it. It was somewhere on a server, somewhere in Minnesota. So we just downloaded it, and we're really the only people playing around with this during Corona times. That was pretty nice. So the first question you ask is, okay, but can we find the standard maps? And we know these V1, 2, 3, these first maps of the brain. Can we find those if we do this connected field modeling? Can we do that using movie watching, for example? And it turns out, I wouldn't be showing you this if it weren't. So we can do this in the retinotopic mapping experiment, and that gives us these beautiful retinotopic maps, right? You see the nice color changes there. It allows us to outline where the colors reverse, and those are the boundaries between the visual regions. So that is, this location in V3 preferentially connects to this location in V1. It shares the color. Yeah? And this is estimated from the retinotopic mapping experiment. You can do the same during movie watching, and you can do the same during resting state, right? So you just get these boundaries back regardless of which experiment you're doing. Yeah? So that's nice, sort of a validation. Yeah? This then also replicates all of those other retinotopic maps that we found in hippocampus in the default mode network, and cerebellum. So you can actually find all of these visual maps during movie watching also. But the nice thing is you can also compare how the brain connects to vision, if you will, during movie watching and during resting state. Yeah, so we now know that we're able to capture this retinotopic organization using this type of modeling. Now we can start to compare between the resting state and when you're looking at something that's actually interesting, right? When you're just thinking in your head, or whether you're actually engaged with a stimulus. Yeah? We can contrast these two different situations. So this is a bit much, perhaps in terms of data, but what this shows is how strong a voxel, a location in the brain, is connected to V1, so how strongly its function is visual in either the resting state or in movie watching. And what you see is that what is red here is these regions that are part of the default mode network. So it looks as if when people are just thinking to themselves, we see that the activations in the default mode network start connecting to vision. It's as if you are generating endogenous images while you're thinking, right? And you need the visual system for that. Yeah, that is a very sort of broad remark, but that's something that I'm thinking might be happening. Conversely, what you see is that during movie watching, you get all of this extra activation or linking to vision in a lot of other regions in the brain. And we know that, for example, auditory cortex is right here. So these are movies with sound. It's when you are connecting the things that you're hearing, people talking, for example, with what you're seeing. It makes sense that that is the type of processing where we need to overlap different sensations, right? And if we're just using vision now, it would also make sense that we see more visually tuned responses even inside auditory cortex, for example. Yeah? All of these lower regions here, they're actually doing things like face processing, processing of scenes, which is exactly the type of thing that your brain is analyzing during a movie that it is not analyzing during one of those simple retinotopic mapping experiments, for example, or during resting state. Right? So this gives us a way of seeing how this visual organization depends on what you're doing in your mind. Yeah. So yeah, this sort of corroborates what we think the brain is doing anyway, but what I really like is this idea that perhaps when you're thinking, your default mode network, that's, you know, very sort of social thoughts, memory related, that you're actually conjuring mental images all the time at that moment, right? And that what your default mode network is doing is it's preferentially connecting to the visual system in this topographic way in order to instate mental images, for example. Yeah, that's all a very wishy-washy thing to say, but I really think that that might be what is going on. Any questions? Great.
Do you think that you end up with an overactivation compared to like most of your default mode network if you were to have some pathology of sorts in the visual center where you have like a quote unquote overactive imagination?
Oh yeah, yeah. So people are the mental imagery field. It's actually a field. They speak of so there's actually a test like for how visual your imagination is. It's like a questionnaire sort of thing. And they say that what's opposite to what you just said, there are aphantasiacs. So people that can think, but their thoughts are as non-visual as they get. So if you try to ask them to imagine something, they just say, "I am sort of drawing light." Right? And so they can think, but it's very non-visual. And so there's actually in the population, there's a broad range of how visual your mental images are, if you will. And I don't think that they tested this in the 180 subjects that I have access to, but that would be a beautiful thing to try out. Yeah, it's a very nice idea. So kudos. I just I can't research it. I'm sorry. Yeah, so I'm pretty sure that this hasn't been done in the literature, the imagination literature. Yeah, but excellent question. Very interesting. I don't know if people, so we could look at, for example, in the psilocybin experiment, you might think that people get more sort of visual in terms of their imagination. We have some resting states, so who knows? That sort of thing might be moving towards answering your question. Yeah. And again, what this highlights, I think, is really what's nice about this sort of cognitive science research is that you can just imagine, you know, how something is, right? You're thinking about, okay, how does thought work, sort of thing, and then a scientific question just pops out, right? I really like that aspect here. It stays very close to how you experience things in everyday life. Any other questions? Because I'm gonna move to something that we haven't published yet, but that I'm like extremely excited about. So if I start talking in a high-pitched voice very quickly, then that's the reason. And I still have some time. So one thing that I've been struck by is that I'm like this written topic mapping is sort of my field. I've done it for a long time, right? Mapping out V1, V2, V3. It's all very nerdy, lovely. But I've also been struck by how shitty it is as an experiment, right? Just making people fixate the center of the screen and then showing them flickering checkerboard. Like you could do that in a cockroach, right? And that would be like essentially the same thing. You're not asking the brain anything interesting, right? And one of the interesting things that I think happens during, for example, movie watching, when you're interacting with a more natural stimulus, is not just this interaction, but also the depth of the experience that you have when you are watching a movie, right? I showed you this, if you do this inter-subject correlation thing, when you do it with people just walking outside, you hardly have any synchrony between brains, right? But in a Hitchcock movie, everybody's doing exactly the same. Their minds are in sync, right? And that's because what's happening there is the director is so good in guiding your entire mind towards specific things, right? And this entire mind thing, it's a very sort of visceral thing. Like a Hitchcock movie is very scary shit, right? And it's a very visceral experience that you get. It's not just visual. That's what I mean by this visceral thing. Just watch this, right? This is actually a movie segment from Inception that was shown to subjects in the scanner during the movie watching experiment. So it's a brief segment, but I think it really highlights this quality of the movie watching experience. Just watch it and try to compare it in your mind with your mental content that you would have fixating the center of a screen and a flickering checkerboard bar just passes by very slowly, right? That's the contrast here.
Very impressive.
You hear anything?
I know this bridge. This place is real, isn't it?
Yeah, I cross it every day to get to the college.
Never recreate places.
Let's try again.
Very impressive.
You see him starting to frown. Right? You can hear this. Certain things are memory.
I know this bridge.
Evidently being shown.
This place is real, isn't it?
Yeah, I
You
cross it every
feel
day
the sort
to
of
get to the
tension
college.
rising,
Never
right? It's
recreate
a
places.
very visceral thing that happens when you watch movies, right? So you actually
Very
experience,
impressive.
in my mind, right, the way that you actually experience bodily sensation. It's just that you're not being touched in real life. It's just that you see someone being touched, you empathize, and then you feel it yourself. Right? You
I know this
vicariously
bridge.
experience these
This
things
place is
through
real,
vision,
isn't it?
just a change
Yeah,
of
I cross
input
it every
channel.
day to get to the college.
Never recreate places.
And so this is something that Nick Hedger has been working on. He's the postdoc in my lab, who's not in my lab. He's actually British, so he's stuck in London for the entire Corona period. But we worked together on this data set because we just had it anyway. And he's been trying to answer this question. He also created the two and three practicals, right? And you can see how that's his thing, because that was also a movie, bodies. You see the sort of theme arising, right? And so we're very interested in this sort of idea of embodied vision. Because when you experience things in a naturalistic context, you don't have a separation between vision, sound, and bodily sensation. They start to fuse. I think you experienced that hopefully that when you saw that conception, and then contrasting it to what a flickering bar would be like. And there's different, if you look at the scientific literature, there's different ways of thinking about how this works. So there's a strong sort of philosophical, ecological bent from the 1950s that calls this embodied cognition, and talks about affordances. So whenever you see a hammer, you don't just see the wooden handle and the metal head, you also immediately experience all the actions associated with that hammer, right? So and these are the affordances that are associated with a visual object. Another thing that people have discovered is these mirror neurons that fire somewhere in the premotor cortex, for example, when you see an action. They also fire when you perform an action. And so they don't care about who's performing the action, whether you're seeing it being performed or whether you're performing it yourself. People have called this mirror neurons. It's a huge discovery. So we thought we would analyze this human techno movie watching thing yet again, and then try to investigate this sort of sense of embodiment that we have using multisensory connected field modeling. And this needs a bit of explanation, I think, so I'll do that in a bit. But the idea is that we try to explain a voxel's responses, let's say somewhere in frontal cortex, not just as a connected field in V1, but also jointly as a connected field in S1. So that's the first somatosensory region of the brain. And so you take brain activation throughout the brain, and then you try to model it from vision, and at the same time try to model it from somatosensation. So it's trying to create a complicated model of what a voxel is doing, and trying to explain it as a function of what's happening in visual terms and what's happening in somatosensation terms. And it turns out that Nick is really smart, and that he was able to actually come up with a way of fitting this. This is how you do it. I won't go into the specifics here, but you can use like complicated regression methods, which are called banded ridge regression. And then you can find a connected field in V1 and in S1 from the same voxel. You get both. And so you get this nice, this is similar to the partitioning that I showed you for this. You'll get parameters on the one hand, and you get cross-validated prediction performance on the other, where you can compare models. This is similarly organized. So this is how information is processed, right? How does something link to vision and to somatosensation? At the same time, you can look at which of these sources of information is stronger in any voxel. So, so you can actually say for each voxel whether its variance is explained from S1 or from V1. So you can say this is a visual voxel or this is a somatosensory voxel. What the is that? It's not like there's a subway passing by or anything. Oh, but we have a poltergeist. Yeah.
What does S1 actually refer to?
Oh yeah, so it's this strip here. It is the so what you see here is the central sulcus. So it's the central sulcus is what separates the frontal cortex from the parietal cortex, the lobes. And so this is the most anterior part of the parietal lobe. It does somatosensation and it contains a full body map. And so what that means is that this patch here, it actually represents like your nose sort of thing, right? And then you have a patch next to it and it represents your upper lip. And then the patch next to that is your lower lip. And so you have like the face here, more or less, then you have your shoulder here, you have hand maps all the way here, then you have like your trunk here and your feet are there. So there's a full body map in that strip. And so it takes in the sensory information from your skin that travels through your spinal cord up almost directly into the somatosensory cortex. Yeah. I think I'll show a picture of that in a bit. And so there's these regions that you've seen in the practical. We call those extrastriate and fusiform body areas. You've seen them pop up in the practical, so we're just focusing on those. They're here and there, and I'm focusing on them here for a reason I'll show you in a bit. Even if you do this motor mapper that I talked about, or we do this separate experiment we have done with mapping fingers and mapping feet, that sort of thing, you see the same region here. So we know that these are interesting regions, right? We already know that they are also connected to bodily sensations, right? Even though they're in visual cortex, and people generally consider them visual regions in the back of the head. They are selected for visual categories, showing bodies instead of something else. And they also activate when you perform actions yourself. Yeah. And so we can perform our analysis and then say first, is a given voxel, is it visual or somatosensory? That's interesting. And you have a clear prediction, of course. Everything that is close to S1, well, it's close to S1, so it's likely going to be somatosensorily organized. Yeah, it's going to also have a similar body map as S1. So we have this somatotopic connectivity instead of retinotopic connectivity there. On the other hand, everything in the back of the brain we think is going to be visual, of course, right? So it's a very clear idea that you would have if you look at this type of result. You would expect the back of the brain to be blue and the top of the brain to be red. Yeah. So when we do this, I'm showing you again the inflated versions here. You indeed see that. Oh, I have to click through. I have some pointers here. You indeed see that like the S1 is on the top, right? And everything next to S1 is red. Yeah. You also see that everything that's like visual in the back of the brain is blue. So that sort of is what you might expect. But if you look at this extrastriate body area, it's red. What this means is that most of the variation in signals that happens there is better explained as a function of a pattern on S1 than it is explained as a function of a pattern on V1. Yeah? So this is really how you would outline extrastriate body area. And it turns out that it is much more somatosensorially organized, if you will, than we would have thought before. And the same goes for this fusiform body area that's there. So it's been stripped there. And so that was kind of surprising to us. We didn't think that we'd be finding such strong somatosensory organization in what we generally would think is a visual region. Now, one thing that's beautiful about this sort of thing is that you can also turn the argument sort of upside down. And what this means is that an analysis like this would only work if there is a consistent pattern happening in S1 that is evoked by visual information. Okay? So this also means that S1 shows structured responses to visual information. Otherwise, you wouldn't be able to find this, right? The source of this pattern is S1. So it means that there is structured information processing in S1 that is actually evoked by a visual movie. It's pretty, there's both sides of the same coin, right? So it's these voxels connect to S1, but this also means that S1 is connected to these regions. And this being fMRI, of course, you now know the caveat, right? We cannot do any fast experiments, right? So if we see a correlation between two regions, fMRI really doesn't allow us to tell which direction this communication is going. So it's just a link. We cannot say what the source of this is, but what it does mean is that you have this entire network of somatosensory organization that is evoked by seeing a stimulus, in a natural stimulus. Yeah?
Find the direction of trenches EEG.
Exactly. Yeah. So then you'd have to like go to EEG or MEG to start to, find this sort of thing. Yeah. And this is exactly, the takeaway also from this sort of, from I guess the entire course also is that if you have a specific question, you need to, choose the measurement modality, if you will, that's optimal for answering that question. And we couldn't do it with fMRI. fMRI is good at spatial patterns. That's what this method depends on. That's what this method finds. We're all good. But if we want to talk about timing, we're in a bind. Yeah. This is clear? So this is our sort of present finding that we're excited about. having shown that there is a much stronger integration between somatosensation and vision than people thought would be the case before. Yeah. So we're trying to sell that idea. And so we can then zoom into this a bit more. And so the next couple of slides are going to be me doing that. And so one thing that you can do is see whether this pattern exists for any type of movie content or for specific types of movie content. Yeah. And so what we decided to do was have a look at whether this pattern of results, this red-blue pattern of results, happens during seeing human actions. And it turns out that that is when you see this pattern. So definitely you get this pattern of somatotopic connectivity with S1 when people are seeing actions. That's cool. Then what we want to do is contrast that to whenever there is no human actions in the movie. And it turns out that there's a beautiful 3-minute segment with just the camera panning through a bunch of scenes where the only living thing in those scenes is a bunch of trees, and at one point there's a horse. But there's no actions of humans involved at all. So you can just do the same analysis in that brief segment of the movie. There's no human actions, and you see that there is no such somatotopic connectivity. So it seems as if this type of response, this type of pattern of connectivity is really evoked by seeing someone do something. So we're trying to get to the bottom. You can look at the difference of points and try to do statistics, but you see that this really caused by seeing the human actions. And so it tells us something about the type of content, the type of cognition that this pattern is evoked by. Yeah, this is not the sort of thing that I'm very much interested in. What I am interested in is these maps, right? I want to go back to the maps because whenever we estimate these connected fields, we don't just get how visual or how somatosensory a voxel is. We also get back the exact patch, the exact location on V1 and on S1 that a given voxel corresponds to. So we could actually start looking into extrastriate body area and project the somatotopic map of S1 into visual cortex, right? The way that we did this before with V1 to the rest of the brain. Yeah. So we have this somatotopic map of body position, like you asked, right? We can actually, this is the way that it's laid out. It's more or less what I just explained, right? And what you get is that you can just assign a given color. So if a voxel in some visual region likes the face, geez, what happened? Oh, yeah, so if a given voxel likes the face region, you color it some color, red or orange, right? And if it likes, I don't know, the feet, you color it purple, and so forth, right? Taking that S1 map and projecting it into the rest of the brain. So this is what that looks like. That again is extrastriate body area. And that again is fusiform body area. And you see that you get this nice map in S1. It goes from red to purple. You can see it at the bottom, top right. It goes from black down to red. It's explaining itself, so that's trivial, right? But anywhere where you see this gradient going from purple through blue to green to yellow to orange to red is where there is a somatotopic map, right? We can just visually inspect this and see these maps. You see that there's a little map-like thing happening here in frontal cortex. Yeah. But more importantly, you see that there's something happening here, right? This is going from red to blue, and this is going in the same direction from anterior to posterior, from red to blue. And similarly here in the fusiform body area. So if we zoom into that, we actually see that there is this band in high-level visual cortex that contains multiple what we call somatosensory homunculi. So there's actually reflections of the body map that we can trace back into visual cortex. So we go from red to blue to red to blue to blue to red to red to blue to red. And so this is like multiple maps that we see in high-level visual cortex. And these are nicely mirror reversed, right? They always reverse in red or in blue. They're also almost identical if you go from left hemisphere to right hemisphere. right? Which is a sort of benchmark. You always need to see that it's bilaterally symmetric. Yeah, so we didn't really know this before. So we're very enthusiastic about this as a sort of finding, like, hey, if you go from, so in this case, like V1 is here, there's V2, V3, and you have all of this sort of bodily organization, even in visual cortex, pretty early on. We're trying to, like, we've been sitting on this for 1.5 years already. Like, the writing of a story and that sort of thing is always the most work. And so the thing that we're finding, the thing that I want to, like, in terms of science, I want to convey here is that conceptually, we think that these maps, these topographic maps, especially of the sensations, are very important in how our brains are organized, much more than we thought before. And I think this also has a lot of repercussions also for how cognition is organized. I like to think, at least. And so, yeah, these topographies we think might be important for this internal alignment, right? To have different brain regions talk to one another, but also because we interact with the world through these sensory organizations. It provides this beautiful reference frame for modeling what's going on, right? In terms of how abstractions are created, for example. And these, you can actually find in functional connectivity that this functional connectivity is structured through these maps, right? So one map connecting to another map, that means that they don't just connect because they're both visual or both somatosensory. They actually, a little patch on one map is going to connect preferentially to another patch in the other map, right? There's very strict connectivity between the two. Yeah, and then you can start seeing this in terms of how your different sensations are blended together when you experience something more viscerally, like this natural movie. And we find these overlaps between the maps, which we think are a really nice way of trying to zoom into how these things also work in cognition. Yeah, and I have this like very pretentious thing about, there's this empirical mantra of like philosophical thing, like nothing is ever in the mind that hasn't first gone through the senses. All sorts of philosophers just keep repeating this. I think Aristotle was first. Anyway, that's the pretentious part of this. And so another thing, this coming from a scientific talk, right, is that I have to acknowledge all of the people that I work with. And so these people are all awesome. And oh, and then you thank people for your attention. Yeah, that's what you do. So that's it. Yeah, don't clap. No. Yeah, thanks for being such a great audience during the entire course. And yeah, I hope this sort of inspired you to at least have an idea of what imaging is like, fMRI is like, and also like where the field is, if you ask me. So yeah, I'll see you guys next week. Great.